I'm confident this program will educate and inspire you, and I want to thank CCTS for taking the lead. So thank you for joining us. I hope you'll participate in the months ahead too, because there's much more to come. So here's to another great program. Now it is my pleasure to introduce Dr. Untitled Chapter, who will open our program today on the exciting topic of constructing a COVID-19 vaccine. Dr. Zhong.
Thanks, Scott, for the very kind introduction. Good afternoon, everyone. Thanks for joining this session. First, I would like to thank OSU CCTS, Corporate Enhancement Office, and the Technology Office of Nationwide Children's Hospital. Specifically, I would like to thank Tanya for organizing this wonderful COVID-19 Inventor Showcase series. I learned a lot from previous sessions. It's our great pleasure to share with you our recent efforts on the development of COVID-19 vaccines. This is a teamwork of many talented researchers, including postdoctoral scholars, graduate students, and undergraduate students. Also, I really appreciate the support from a number of OSU core facilities, such as the imaging facility, flow cytometry facility, and the genomics facility. They provide huge support, especially during the challenging time of the university lockdown. Now, I would like to introduce our next speaker, Dr. Chongxi Zhong. Dr. Zhong is a research scientist at the College of Medicine now. He was a postdoctoral scholar in my lab, working on messenger RNA engineering for over 4 years. In early 2020, he made significant progress in improving mRNA translation efficiency. Then the COVID-19 occurred. We thought maybe we can apply our new RNA technology to construct a COVID-19 vaccine. Dr. Zhong will introduce the details of our work. Let's welcome Dr. Zhong.
All right. Thank you very much, Dr. Dong. Thank you for your kind introduction. Again, my name is Chongxi Zhong. I'd like to give you more details about how we construct this COVID-19 vaccine. So the general scheme of this work was we started with optimizing the 5' and 3' untranslated region of mRNA, the UTRs, mostly following two rationales. The first is endogenous expression analysis. In this one, we try to identify the top performing endogenous UTR candidates. Then we designed new UTR de novo. So eventually, we compare the best candidate from these two rationales and identify the optimal one. Then use this optimum UTR to synthesize mRNA that encode the antigen of SARS-CoV-2. After that, we use lipid nanoparticle formulation to deliver it. Then analyze the expression of antigen in vitro and also in vivo in mice. After performing vaccination, we analyze the antigen expression and also the antigen-specific antibody production in mice serum. So the first was to identify the top performing endogenous UTR. And in order to do that, we thought we need to calculate the protein production capacity per mRNA molecule. And we need two numbers in order to calculate that. The one is mRNA half-life. The other one is translation rate constant. So with these two numbers, we can use this equation and do a very easy calculation so that we can get how many copies of protein were produced by each mRNA molecule throughout its half-life. And because nowadays there are a lot of omics data, and we really didn't need to reinvent the wheels. So we did literature search and found these two numbers from literature. Then we did calculation and the ranking. Overall, over 4,000 mRNA were ranked according to their protein production capacity. And this RPS27A was ranked the highest, several-fold higher than the second best one. This was interesting But we had one more thought. Because the length of protein varied dramatically from several dozen to several hundred or even thousand amino acids. So we thought, could this number of protein underestimate the protein of different length? So we did one additional calculation that factored in the length of protein. So this number, this protein copy per mRNA was from the previous page. Then we multiply it by the number of amino acids in each protein. Eventually, we get how many total amino acids were produced from one mRNA molecule throughout its half-life. Then did the racking again. Still, this RPS27A ranked the highest and still about threefold higher than the second largest one. So this was very interesting to us. Then we start to pay a close attention to this RPS27A gene. And this is a housekeeping gene that encodes one component of the ribosomal 48 subunit. And from the database, we can see there are two protein coding transcripts. They both encode the same protein, but use different UTRs. And they actually use two different 5' UTR. One is longer, we name it 45. The other one is shorter, we name it 44. These two numbers were based on the last two digits of their transcript ID. And these two transcripts share the same 3' UTR, we name it S27A. And a further close analysis of this UTR reveals that these two 5' UTR actually contain confirmed or putative top motif, the terminal oligopyrimidine motif. That's a motif reported to repress mRNA translation and certain stages of the cell cycle. So that's not something we want. Therefore, I designed two new 5' UTR with these two underlined top motif removed. And we name them 45' and 44'. So totally here we have 4 mRNA sharing, using 4 different 5' UTR, but share the same 3' UTR. And we start to analyze their translation capacity, their kind of translation efficiency, using firefly luciferase coding region as a reporter gene. So the result you can see here, in general, the one with shorter 5' UTR, this 44, perform better than the one with long 5' UTR. And removal of this top motif further enhanced the translation efficiency. And also, throughout this optimization process, we used several controls obtained from literature, including the UTR from CYBA gene, and also UTR from this AG alpha globin gene. AG plus G means the alpha globin with one additional G to make a complete Kozak sequence. And also this 5 AG plus G means this AG plus G with only 5' UTR without any 3' UTR, and also one no UTR control. So as you can see, this 44' performed the best, and all the results were normalized to CYBA. So this performance was about 1 to 1.5 compared to this CYBA. Then for 3' UTR, I use the same 44' 5' UTR and compared 4 different endogenous 3' UTR, including this S27A and also alpha-1 antitrypsin and transferrin. These two are abundant serum proteins. And also we tested one additional 3' UTR of HCV viral RNA, because according to a publication from Jennifer Doudna, this 3' UTR could recruit 48 subunit of ribosome and facilitate and accelerate reinitiation of translation. So we hope it can kind of help our application. But in our assay and our experiment, they all perform very similar. So eventually we still choose this S27A as our endogenous 3' UTR because it's really short. It has only 34 nucleotides. And the other 3 range from 100 to near 400 nucleotides. So with this endogenous candidate, we started to design new UTRs de novo. After literature search, I decided to use 3 major criteria. The first one is to use the Kozak sequence, this GCC-ACC, because this is a known sequence that facilitates the recognition of translation start, AUG, by the initiation complex. So as discovered by Kozak. Then I need to avoid upstream AUG because this can induce unwanted upstream translation start. And in worst case scenario, it can even induce the frame shift mutation. And the third criteria was I need to minimize the secondary structure because the common understanding of translation start was the scanning mechanism. So in this mechanism, the translation initiation complex scans through the 5' UTR until it finds the optimum AUG to start translation. And the secondary structure in the 5' UTR can slow down this process because the complex needs to pause and use energy to unwind the secondary structure so that it can go through it. So with this criteria, one open question remains, how long should I design this 5' UTR? So we decided to find out through designing of 5' UTR with different lengths, all following these 3 criteria, but with different lengths, ranging from 10 to 90 nucleotides. And the result was interesting. So the translation efficiency actually increased along with the length of 5' UTR until you go beyond 70 nucleotide. So the optimal length of 5' UTR determined in this work was 70 nucleotide. Then we perform another analysis, nucleotide composition analysis, in order to find out in this 70 nucleotide 5' UTR, what was the optimal nucleotide composition. So I designed a total of 8 different constructs. So from 1 to 4 here, you can see they have various number of A, C, and G. But most importantly, they didn't have any U. The rationale was when we designed this one, and throughout this work, we were using 100% pseudouridine modified mRNA. And we suspect that incorporating too many pseudouridine in 5' UTR may impair translation because naturally there aren't that many pseudouridine around. Then as a comparison, also I designed 4 additional with increasing amount of pseudouridine from NCE 5 to 8. And the result was not something we expected initially. So without pseudouridine, the translation efficiency dropped. And with increasing amount of pseudouridine, they either remain the same high efficiency or increase the efficiency along with the U content. So here, we decided to choose these NCE 7 and 8 as our candidates for further optimization. So for 7 and 8, I analyzed if they have microRNA binding site in 5' UTR. Common knowledge for microRNA, everybody knows, mostly microRNA target mRNA and 3' UTR. But there are some publications showing some microRNA target mRNA and 5' UTR. So I analyzed this NCA 7 and 8 for their potential microRNA target site. And indeed found some putative ones. So I removed them and made 7D and 8D. And this 7D indeed showed slightly enhanced translation efficiency as compared to its original form 7. So we decide to choose this 7D as the optimal design, a de novo design 5' UTR. Then how about 3' UTR? What de novo design can we do? So there wasn't much to learn from literature. So after a lot of readings, we came up with the idea, which is to add functional motifs to this S27A 3' UTR. And in previous de novo design of 5' UTR, I always use the same S27 because we need to have something there. So now we want to add a functional motif to make it better. The rationale was because in 3' UTR, some natural 3' UTR are really, really long, like up to 10,000 nucleotide. And they have a lot of regulatory elements in those regions. And nowadays, the field doesn't really know a lot about their interaction, their rationale, but just kind of some group reported individual studies on specific element. So mostly there are two major categories. One is a microRNA that mostly repress translation by mediate degradation and repression. And also another one is RNA binding protein. Although some was reported to have negative regulation, but indeed there are quite a few had showed positive regulation. So we obtained those candidates from literature. Two RBP, RNA binding protein candidates. One is QKI7. So it was reported to induce cytoplasmic polyadenylation. So basically it can add A to mRNA in cytoplasm. Very exciting to this mRNA application. So I obtained two binding motifs for this RBP, the quicker response element 1 and 2. And the other RBP candidate was HUR. And it was reported to bind to the transcript and stabilize it and enhance translation. So I obtained two binding motifs. One, AU-rich element from endogenous human mRNA, and the other one from Symbus virus. This R3U stands for repeated element 3 with uridine-rich element. I name it R3U. And also we obtained the fragment of IRES from two viruses, because these two IRES were reported to recruit 48 subunit of ribosome, and we hope it could facilitate translation reinitiation. And the result, mostly were negative. So you can see the control is no motif, which is the S27A alone. So the two motifs for QKI7 didn't work, and the IRES fragment didn't work. But fortunately, both motifs for this HUR worked. And the one from virus, this R3U, worked better than the ARE. So we decided to choose this S27A plus R3U as the optimum de novo designed 3' UTR. And you can see the performance of this de novo designed UTR was actually much better than endogenous UTR candidate. Here, over CYBA, it's about 5-fold, and previously endogenous was only about 1 to 1.5-fold. So that means the optimum UTR combination was NCA7D as 5' UTR, and also this S27A with R3U as the 3' UTR. And we select NASAR, NASAR as the code for this optimum UTR combination. So to further benchmark the performance of this NASAR UTR, we did more literature search and found more UTR reported in literatures. We named them MOD1 and MOD2 because they are used by nucleotide modified mRNA, and MOD means modified. But later, after publishing this work, I realized the MOD could actually be an abbreviation for Moderna. I promise we didn't plan to mean that, but it wasn't misleading either, because this MOD2 was actually indeed Moderna's UTR sequence. So they use it in their Zika virus mRNA vaccine, and also a bunch of other applications, including the current and the very famous COVID-19 mRNA vaccine. So as you can see, as a comparison, this NASAR mRNA performed really well and better than all the controls we tested in this work in cell line, HIP3B, 293T, and also in primary cell, bone marrow derived macrophage. Then with the encouraging result in UTR optimization, we started to develop this SARS-CoV-2 mRNA vaccine. We synthesized two mRNA using this NASAR UTR to encode either RBD fragment or the full-length spike of SARS-CoV-2. First, we need to analyze their expression and after switching the coding region. So we use the RBD encoding mRNAs with different UTRs. And still, the performance remains the same. NASAR UTR outperform other controls, very consistent with what we saw from luciferase coding region. Then we also analyze the expression of these antigens on cell line using immunostaining. So you can see this RBD was stained as green, and the spike was spiked as green also. So after confirming their expression, we start to do the in vivo experiment. The first was to determine what lipid nanoparticle formulation to use. And we tested two. One is a TT3 lipid nanoparticle developed by Dr. Dong. And then the other one was the MC3, a FDA approved LNP formulation. So as you can see, when we use the luciferase mRNA and perform the in vivo imaging, the iris imaging, the TT3 nanoparticle induced much higher, over 2 orders of magnitude, higher luciferase signal in mice, which was encouraging. Then we also compare the formulation, but this time with actual vaccination in mice. We use the spike encoding NASAR mRNA, and the y-axis here shows the spike-specific serum IgG in mouse serum. Then the result was consistent with luciferase, and totally over 300-fold more spike-specific antibody was induced by this TT3 NASAR mRNA vaccine. And we also performed, compared different injection method, and intramuscular injection was found to be several-fold more efficient than subcutaneous injection. And we use the same RBD encoding NASAR mRNA as revealed by ELISA. So eventually, we performed a prime and boost vaccination in mice. So each mice received 2 injections. We used the TT3 lipid nanoparticle and performed intramuscular injection. On the x-axis here shows the dose of mRNA, the microgram of mRNA mice received at each injection. And also we compared these two mRNA vaccine, the spike encoding and RBD encoding. As you can see, the serum IgG level specific for that antigen was very high and high dose level. And I suspect maybe there was some kind of saturation of immune response. And lower dose level, the spike encoding mRNA appeared to be more immunogenic. So overall, in this study, we identified the optimum performing NASAR UTR that can enable the high expression from mRNA. And this NASAR UTR was used on this COVID-19 mRNA vaccine and showed robust performance. And in our understanding, we hope this UTR can be, we think this UTR can also go beyond this, can be used in other mRNA-based applications. So with that, I'd like to thank Dr. Dong for his great guidance and leadership in this project. And also I'd like to thank the strong support I received from other lab members, especially Xucheng Hou, because I couldn't finish all the work myself. And the funding from this work was from NIH and OSU College of Pharmacy. And OSU has already filed patent for the NASAR UTR and COVID-19 mRNA vaccine developed in this work. And last but not least, we really appreciate the fantastic equipment and services provided by several core facilities on campus, including genomics, microscopy, and flow cytometry facilities. And with that, I'd like to hand it over to Dr. Paul Stoodley, the Director of Campus Microscopy and the Imaging Facility, and he will bring you more details.
Hello, good morning. My name is Paul Stoodley. I'm a Director of the Campus Microscopy and Imaging Facility, which also serves as the OSU Comprehensive Cancer Center Microscopy Shared Resource. Looks like the slide is having a bit of an issue. Hopefully it'll sort itself out. I'm a professor in the Department of Microbial Infection and Immunity, as well as in the Department of Orthopedics. In the campus, in the CMIF, Tong Zhang is our associate director, and he's an expert in super resolution microscopy and confocal microscopy. Brian Kemino is a expert in confocal microscopy and electron microscopy, and Jeff Tonigs is an expert in TEM and sectioning. And then we have various folks assisting with finance and IT. I'd like to point out Dr. Alan Sung and Richard Fischel. They're co-directors of the Microscopy Shared Resource. I'm also a member of the Infectious Disease Institute, and if we go to the next slide, I can just give a little bit of a shout out to the Infectious Disease Institute that Tanya asked me to briefly mention. So the IDI is a network of researchers across OSU that are looking at various aspects of infectious disease, whether it be virus, bacterial, parasitic, and how that affects a multitude of different human activities, all the way from health to food resources to veterinary concerns. If you'd like to find out more about this research, I urge you to, or this resource, I urge you to go to the OSU, Google OSU and Infectious Disease Institute, and then you'll find a website which which shows you the resources available, and also kind of breaks down into the various focus groups of this very ambitious and productive project. Okay, if we go back to the next slide, I briefly want to talk about the CMIF. So our mission of the CMIF is to facilitate research. This is generation of papers, proposals. Basically, we have two components. We have the equipment. We have all the bits of kit, all the microscopy expertise that we allow, that we bring to OSU. But I think a very big part of what we do is the people. And so the people are the folks that have the expertise that can assist you with your experimental design, etc. So our various microscopy, we have a range of confocal and light microscopes. CMAS houses cryo-EM, very exciting for doing atomic resolution microscopy, as well as having electron microscopy. When we go to the next slide, I just want to highlight our new instrumentation. We're just in the process of installing, should be done by the end of November, our new Nikon SIM Storm system, which is equipped with turf, where we can go from confocal, which is about 120 nanometers. Now we can get down with Storm to 10 nanometers on fully hydrated specimens. So we're really excited about rolling that out. Okay, if we go to the next slide again, thinking about sort of going forward, OSU, the mission of the CMF is to keep OSU on the cutting edge research with advanced microscopy techniques and equipment, provide educational seminars and workshops. Our users, we're based on training our users to use the microscopes. So that's education is a very big, important part of our mission. We're also collaborating with various shared resources on campus, as well as CMAS, where we're looking into bringing in correlative electron and light microscopy. So in the last slide, this is a very brief overview. If you do use us, please look us up on the website. We'll be happy to talk to you about your experiments, happy to go over one-on-one the instrumentation and our expertise. And if you do use us, please let us know what papers and grant proposals we helped with. These metrics really help us to go to the Cancer Center Office of Research and provide our impact that we have to OSU. With that, I'll wrap up, and I'd like to introduce Dr. Jeffrey Chalmers, who's going to be talking about flow cytometry.
Greetings. I'm here to give you an outline of our flow cytometry shared resource. In particular, as I was introduced, I'm Jeff Chalmers. I am a professor of chemical engineering, chemical and biomolecular engineering, as well as director of the shared resource. And also within the last 6 months, the Cancer Center has hired Kevin Weller, who is basically an expert on not just flow, but using the new fluorescent spectral deconvolution flow cytometers. And in particular, his specialty is also with the new Cancer Immunological Institute. All right. As you can see, our aims, this was put together for our cancer grant renewal, but I thought it does a nice job of summarizing our focus of our core. So we're here, obviously, to provide state-of-the-art equipment. We are constantly working with the CCC membership as well as anyone else that comes to us. I want to make that clear that while we're a CCC core, we are here for the whole community. And we train people such that you can use the facility 24 hours a day, not just when our people are here. Okay, in terms of our services, here on the right, you can see we definitely consult with people, help them design their experiments. We can do self-sorting, obviously analytical cytometry. We have connections to the mass cytometer on campus. I personally am interested in circulating tumor cell work, so I do that with people a lot. We are further developing our exosome analysis techniques, and I hope within the next year or two to really have more new technology and expertise in that area. And all right, now let's move on. In terms of equipment, you can see here we have a number of BD instruments, both analysis and sorting instruments. We have a Cytec Aurora, which is that instrument I referred to that does spectral imaging flow cytometry. And we're working on getting sorters that are based on that. All right, let's move on here. Just a couple of examples. We always like to highlight high impact papers we do. And I want to show you in this one in particular that it's not just flow we're doing, but we do in conjunction with our researchers and nanoparticle work. So we're actively involved with other shared resources as well as new technology. Another paper here just showing how we're interested in microRNA analysis going into cells. And just another one, which I can talk to you later about. Okay, so maybe to summarize, we are working on further developing new equipment, bringing exosome research, as I mentioned, and we really are excited about the new potential of high number of marker sorting, which will go with these new spectral deconvolution instruments. All right, my turn now to introduce Amanda. And there she is.
Yes, so good afternoon, everybody. I am Dr. Amanda Toland. I'm co-director of the Genomics Shared Resource, part of the Comprehensive Cancer Center's many shared resources. And the goal of our GSR is to really support high quality genomics expertise and technology in order for researchers in the Cancer Center and across OSU and nationwide to get top-notch data. I'm going to introduce to you our team on the next slide. So we have individuals in our team at OSU on the second floor of the Biomedical Research Tower with expertise ranging from nanostring, gene expression, Sanger sequencing, and next generation sequencing of libraries. And then on the right, you'll see our team at Nationwide Children's, and they are the big experts on doing the high throughput next generation sequencing. I would like to go over some of our services on the next slide. We offer many different technologies and services, including next generation sequencing, many different types of library design, preparation, and QC of those libraries. We have DNA and RNA quality control measurements that we can do for you. There's a number of different technologies that we can use to assess transcript and microRNA expression profiling, including RNA-seq, single-cell RNA-seq, nanostring digital counting, Affymetrix microarrays, and digital PCR and qRT-PCR. We also offer conventional Sanger sequencing, as well as genotyping of SNVs and microsatellite genotyping and cell line verification. We have a number of different quantitative PCR-based assays, including DNA copy number, structural variant, and SNP genotyping. But one of the things we really stress is that we are there for you to help in experimental design, help you navigate the complex array of technology choices there are to choose the one that's best for answering your research questions. We're also there to help in grant writing and paper writing. There are a couple of new instruments I'd like to highlight, including our 10x genomics chromium controller for single-cell RNA-seq, a cell-seq genesis instrument, which enables us to do single-cell NGS applications for full transcriptome. And at Nationwide, there are 2 NovaSeq 6000s. There are also a number of pieces of equipment that we are just bringing online, including our NanoString GeoMX digital spatial profiler, which we just got installed. We have not yet been trained on this, but we hope to offer this soon. And we also have a PacBio Sequel and Sequel 2. On the next slide, we'll highlight some more of the instruments, including our DNA analyzer 3730s, which are used for the Sanger sequencing, and our NanoString nCounter. As I mentioned, we have 2 locations, one at the OSU campus in the Biomedical Research Tower on the 2nd floor, and our partner lab is at Nationwide Children's. And with that, I'd like to introduce the co-director of the Genomic Shared Resource, Dr. Richard Wilson, who will continue to describe.
Thanks, Mandy, and good afternoon, everybody. I just wanted to follow on to Mandy's point. With a focus on our sequencing capacity, most notably the addition of 2 Illumina NovaSeq instruments that are now up and running here at the Nationwide Children's site. Pretty excited about the ability to do work on these. There are 4 different flow cell configurations for running samples. And with the most dense of these, we're actually able to sequence quite a few human genomes to 30x coverage in single runs. This allows us now, besides doing whole genome sequencing, genomes from non-human organisms, RNA-seq analysis, etc., we're able to offer now a rate of $985 cost per human genome. This is, of course, contingent on being either an NCH investigator or a member of the Comprehensive Cancer Center at OSU, because these 2 entities subsidize some of the cost. But if you have at least 24 samples for human whole genome sequence, we're now able to do this for under $1,000. And then if you'll go to the next slide, we just want to highlight some of our upcoming future plans and services. We have quite a bit of work going on for single-cell DNA and RNA sequencing. We have quite a bit of experience now with long-read sequencing technologies, most notably, as Mandy mentioned, using the Pacific Biosciences Sequel and Sequel 2 instruments. This has now gotten to the point of being fairly routine and reliable. And we have some nice software tools for the assembly and the merging of long-read data with short-read data. We continue to do quite a bit of work on single-cell RNA sequencing and analysis protocols for that. We're developing new methods for looking at circulating tumor cells, full-length transcriptome analysis, as well as developing protocols for nanostring and the GeoMX digital spatial profiling that you heard Mandy mention. You can contact either Mandy or I for more information. Our emails are shown on this slide. And I'll take that cue then to introduce you to Carolyn, who will continue on with this presentation.
Great, thanks, Richard. My name is Carolyn Crisafulli, and I am the entrepreneur in residence for the CCTS. And we'd like to open this up to questions from all the participants. We've got a number that have come in. If you have any additional questions, please go ahead and post those into the Q&A. Those questions that we don't get to, we'll be sure to respond in emails. So the first question I've got here is for Dr. Zhong. Were the RNA stabilities measured before the transfection?
So the question was the RNA stability performed before transfection?
Measured before the transfection.
Before the transfection. So before the transcription, it's still in test tube. So yes, we did measure the stability. So in that case, we used the bioanalyzer in genomics facility to make sure all the mRNA we synthesize before use had the same quality. Essentially, I think that's the quality control question. Yeah, so we did assess the quality of the mRNA by bioanalyzer. And they are pretty good.
Great, we have another one kind of along that line. Have you measured the half-life of these mRNAs flanked by your UTRs in circulation or in various tissues in the mice?
Not in mice, but we did try to measure the mRNA half-life. But in our assay, they seems to have the same half-life. I guess we still need more work to kind of determine that. From the data we have at this point, there's no dramatic difference in half-life.
So there's a follow-up with that. If that's not been tested, can you speculate on the half-life of your mRNAs and also the vaccines, perhaps that Moderna and Pfizer are making?
Half-life, based on our data, they are similar, very similar. There's no big difference. So basically, in our assay, we compare their kind of expression and also half-life side by side. There are kind of big difference in the expression level, but half-life wise, we didn't see big difference.
Okay, let's see here. Thanks for those answers. Let's see. How about, have you had a chance to monitor how long the IgG level lasts?
In our prime and boost vaccination, we did one injection in the beginning, another injection 2 weeks later, then analyzed the IgG level and week 4. But we didn't test them after that because the mice were sacrificed.
Okay, let's see, another one we have. How did you measure the mRNA half-life?
Well, so that was not part of the kind of data shown here, but we just test the expression level and different time point. Yeah.
Okay, let's see. If, please feel free to share any others. Let's see what else we have. We've got a lot of questions here for Dr. Zhong. Let's see. So what are going to be some of your next steps? And I mean, this is a very lengthy process to develop a vaccine. What are the next steps that you see for the project that you're working on, and then additional resources then that may need to be drawn in as you move this project forward?
Yeah, this is a very good question. So basically at this point, because we are mostly doing mRNA sequence engineering, then apply this discovery, this NASA UTR in this COVID-19 mRNA vaccine. At this point, we lack the capacity to move it forward towards clinical. So I like to let anybody who's watching know if there is any opportunity of collaboration, we'd like to maybe move this forward. And also, please contact Dr. Dong and me. We can have some discussion.
Elaborate on that. What types of collaborators would be ideal for you?
Maybe can we bring up Dr. Dong?
Yeah.
I'd like to hear more opinion from Dr. Dong.
I think Chongxi mentioned, so this is a platform technology we developed. It can be applied to quite a different and diverse therapeutic applications. Right now in our lab, we are working on different projects and for treating cancers, for treating other type of infectious diseases. So this one can be a very powerful tool for future applications.
In a variety then of disease states and infections then, is that right?
Exactly, yeah. Yeah, we can also work this on bacterial infections, not just viral infections.
Great, great. Let's see. We've got another question here. If someone gets a Pfizer vaccine, how long do you think the mRNA will survive in the body? And how long might that compare to what you are proposing?
In general, for mRNA-based therapeutics, the mRNA doesn't last very long, typically 1-2 days at maximum. But the protein expressed by mRNA can last longer, and that need to be determined case by case. But for other vaccines, we really couldn't tell how long they could last. But that's a general scheme for all the mRNA-based applications. Yeah, especially for this COVID-19 mRNA vaccine, when we express the antigen, this antigen can be taken up by the antigen-presenting cells, then induce the systemic effect throughout the body. So the effect can last longer, but the mRNA itself, then the antigen itself, doesn't necessarily last very long.
Okay, great, thank you. So I've got a couple.
I would like to.
Oh, please go ahead.
Yeah, I would like to add one point. I think you are asking what type of collaboration we are looking for. Currently, our technology office really helping us to move the project and technology forward to real applications. Several venture capitals and biotech companies are working with us. We would like to move towards to the clinical trials. So if anyone in the audience or in the community would like to work together for any type of collaboration or resources to move this, such as scale up this product or initiate some R&D and help with phase 1 trials, because this one can be applied to different type of therapeutic applications. I think we have broad interest working with different collaborators.
And how may they reach out to you? Do we have a slide with your contact information, Dr. Zhong and Dr. Dong?
Let me type into the chat. So my email is dong.525@osu.edu. If anyone is interested, feel free to email me.
Great, great. Okay. So we've got time maybe for one more question. If anyone has got a Oh, great. Thanks for sharing your email address. So everyone's able to see that. One more question here, and then we'll wrap it up. Got a question. Would you like to test your mRNA in attenuated virus vectors? We've got 2 here on campus.
Yeah, that would be wonderful.
Yeah, yeah, we'd love to do that. Because that really tells kind of the neutralizing ability of this mRNA vaccine.
Terrific. So we, you know how to reach Dr. Dong now. So that's excellent. And this is really what we are, the impetus behind this program is to bring collaborators together from different areas across not only the university, but the community. And we really appreciate you asking the questions and sharing. We had a terrific panel today with the resources that are available. And we like to show that each time to understand the breadth that's available here at the university. So with that, I'd like to wrap up. Let me share my screen.
Thanks so much for having us.
We'd like to thank our, here we go. Sorry for the delay here. So I want to thank our panelists, Chongxi Zhong, Izhou Dong, Paul Stoodley, Jeffrey Chalmers, Richard Wilson, and Amanda Toland. Terrific presentation today. We'd also like to thank our sponsors. This monthly series is co-presented by the Ohio State University Corporate Engagement Office and Nationwide Children's Office of Technology Commercialization. We've got a growing list of mission partners. If anyone is interested in further engaging with the CCTS, please reach out to Tanya Matthew. And please, we'd like to give you a warm invitation to join us next month for our COVID Inventor Showcase. This will be on November 17th, again, 12 noon to 1 o'clock. And this is going to be featuring Dr. John Bird, Jennifer Boyack, and Zainab El Baghdadli. From the Wexner Medical Center, Comprehensive Cancer Center. And the feature of this program is going to be a BTK inhibition for therapy of COVID-19. So with that, we'll give you a couple of minutes back on your day, and we hope to see you next month. Please feel free to reach out to any of the speakers today, as well as the resources with any additional questions that may come up. And we would really like to welcome you to seek out additional collaborations with those that we have featured today and in our upcoming sessions. So thank you all, and we look forward to seeing you next month.